Ever wondered how scientists replicate a specific gene? Want to know more about gene cloning? Well, you're in the right place. Gene cloning. It sounds like something out of a science fiction movie, doesn't it? But it's very real and plays a crucial role in the realm of biological research. So what is gene cloning exactly? It's a process where a specific gene of interest is located and copied out of all the DNA extracted from an organism. It's like finding a needle in a haystack, but with the precision and accuracy that only science can provide. In this video, we're going to explore this fascinating process. We'll introduce you to the world of gene cloning, breaking it down into easy-to-understand steps from DNA extraction to the creation of gene libraries. Ready to dive into the fascinating world of gene cloning? Keep watching! The process of gene cloning might sound complicated, but it's all about following a series of steps meticulously. Let's dive into this fascinating process. Our journey begins with the extraction of DNA from an organism known to have the gene of interest. This DNA is then cut into gene-sized pieces with something called restriction enzymes. Think of these enzymes as little molecular scissors, snipping the DNA into the right-sized fragments. Next, we turn to bacterial plasmids. These are small circular pieces of DNA that bacteria naturally exchange. Interestingly, these plasmids are cut with the same restriction enzyme. This ensures that the cut ends of the plasmid DNA match the cut ends of the gene-sized DNA fragments. Once cut, the gene-sized DNA and the plasmids are combined in one test tube. Here, a phenomenon occurs. The plasmid and gene-sized piece of DNA often anneal together, forming what we call a recombinant plasmid, or recombinant DNA. This is the essence of gene cloning, creating a new piece of DNA by combining two different ones. The recombinant plasmids are then transferred into bacteria using methods like electroporation or heat shock. It's a bit like sending the plasmids on a trip into a new home. Once comfortably inside the bacteria, these plasmids get to work. The bacteria is plated out and allowed to grow into colonies. Each of these colonies is a little factory, producing more and more of the recombinant plasmid. All the colonies together form what we call a gene library. The final step is the screening of this gene library. The goal is to identify the colony containing the gene of interest. This is done by looking for either the DNA sequence of the gene, the protein it encodes, or a DNA marker close to the gene. So that's the magic behind gene cloning, but let's summarize what we've learned. Gene cloning is a fascinating process, isn't it? Now, let's refresh our minds with the key points. The journey begins with extracting DNA from an organism known to possess our gene of interest. This DNA is sliced into gene-sized pieces with restriction enzymes. Meanwhile, bacterial plasmids are cut with the same enzymes. These two components are then combined, often forming a recombinant plasmid. This recombinant DNA is then integrated into bacteria using electroporation or heat shock, and these bacteria are allowed to grow into colonies, forming a gene library. This library is screened to identify the colony containing our gene of interest. Now, here's the twist. Antibiotic resistance genes naturally present in bacterial plasmids used in gene cloning help genetic engineers to screen out any non-recombinant bacteria. That's a wrap on gene cloning. Don't forget to subscribe to our channel Digianalix for more insightful videos. Stay curious, stay informed.